Our final speaker of this session is Professor Charlie Serhan. I think that Sir John, if he was sitting in the audience, would be very, very excited about this, because I think what Charlie's done is opened a completely new area for lipid research, and I think he would very much enjoy sitting in the audience and listening to what you have to say. Charlie's going to tell us about elucidation of resolution mediators in infectious inflammation that signal regeneration and bacterial clearance.
Okay, well, thank you very much for the kind introduction. Can everyone hear me? It's okay on the audio. It's a great pleasure for me to be here to help celebrate the 30th anniversary of the William Harvey. I'm a great admirer of pharmacology and the research that's gone on here at the William Harvey, and certainly a big admirer of Sir John Vane. And it was already back in 2008 that I had the opportunity to give the Sir John Vane lecture. Time really flies. So I want to be able to tell you what we've been doing since then. My institution requires this disclosure, suffice it to say, from our basic research, supported by NIH. We had an opportunity to the scientific founder of Resolvin Pharmaceutical. Their mission was to take some of this work to the clinic. I should point out I have nothing to do with the company today. I think inflammation is certainly close to all of us. We're all impacted by acute inflammation. On this slide, we started to think about the decision paths for acute inflammation, chronicity or resolution. Obviously, the ideal outcome is resolution. So, as you know in the textbooks, resolution of acute inflammation was thought to be a passive process, meaning what? Meaning that the inflammatory mediators on the initiation of an acute response would just dilute and dissipate. And what we learned studying self-limited acute inflammatory responses, which we really learned from this gentleman here, Derek Willoughby, befitting that we're in his lecture room today, that self-limited responses give rise to the active process of resolution, where resolvins and other SPMs, a superfamily of pro-resolving mediators that I'll tell you about today, take part to bring us back to a state of resolution and homeostasis. Now, we have focused our efforts on periodontal disease over the years because this is an acute, it's a chronic inflammation, but certainly a big public need in the United States and around the world. And this is a patient of my colleague Tom Van Dyke. And this is a very good example of leukocyte-mediated tissue destruction around the ligaments of the tooth. So we thought that this would be an ideal place to test the principles that I'll talk to you about. So today's outline, we're going to emphasize our question that we're really interested in. What controls excessive inflammation and infection? How are these linked together? And give you an update on the specialized pro-resolving mediators, which each of these are a separate family of bioactive mediators, the resolvins, protectins, Marisins. And then I want to introduce you to new pathways that we've identified that play a role in tissue regeneration, and talk about the stereochemical assignment via total organic synthesis of these mediators. So I'd like to point out this illustration that we were able to put together from our work, and it emphasized the resolution of inflammation as an active process with novel mediators and controlled mechanisms. Now, as you'll hear from Professor Samuelson's talk later on today, his seminal work on the discovery of the biosynthetic pathways for the prostaglandins and leukotrienes. These are very important mediators, as you know, in evoking the cardinal signs of inflammation. And I'll point out that they play a critical role at the beginning of the acute inflammatory response, and that in controlled laboratory setting, we've been able to put together this temporal sequence of events to the signs of resolution, which are regulated by the pro-resolving mediators. There is a lipid-mediated class switch that leads to the production of the lipoxins and the resolvins that gives signals to macrophages and the non-phlogistic recruitment of monocytes, and they eat and take up the apoptotic neutrophils. When added back pharmacologically, the resolvins can regulate and counter-regulate prostaglandins and head off chronic inflammation in animal models as well as organ fibrosis. And the Marisins and the Resolvins stimulate resolution of wound healing. So the new concept that I'd like you to think about and take home today is that at time zero, experimentally, the resolution process begins, and it is an active process. So the two basic biological responses that we use for structural elucidation of the pro-resolving mediators is the cessation of neutrophilic infiltration, the blockade of diapedesis, to limit further neutrophilic infiltration to a site of inflammation, and this is the physiologic response at the postcapillary venial. When added back pharmacologically, this is perceived as anti-inflammation. And this is quite different than the pro-resolving action, which is actually comprised of both of these, where the pro-resolving mediators increase the clearance and killing of phagocytosis and phagocytosis of the apoptotic PMN. So it took us quite some time to recognize that pro-resolution was not identical to anti-inflammation. And to be able to do this systematically and gain the evidence, we introduced resolution indices to be quantitative in experimental animal models. And this extended on to, our surprise, the uptake and killing of microbes. And I'd like you to keep that in mind in terms of thinking about immune suppression. So systematically, within the resolution phase, we were able to assign the stereochemistries and biosynthesis of the E-series resolvins, illustrated here. There are three of them, resolvin E1, 2, and the third was born in Tokyo, former fellow Makoto Arita. Docosahexaenoic acid, as you know, is enriched in the brain and in human testes, and DHA we found is also used by inflammatory exudates, pus if you will. It's transformed to D1, 2, 3, and 4, all of which we've carried out the complete stereochemical assignments. During this process, this hydroperoxide intermediate gets converted to an epoxide intermediate, which is precursor to the protectin family, and neuroprotectin D1, which we've worked on with Nicholas Bazan and his group. And then later on in the inflammatory response, macrophages arrive in the exudate and they convert DHA to the Marisin family, illustrated here. So the resolvins, the lipoxins, the protectins and Marisins have been studied by a number of laboratories, and as illustrated here, in nanogram to microgram amounts, they are stereoselectively active. They have a novel mechanism of action. They're active in the airway, cardiovascular, ocular, in renal function, within the brain, periodontal, which I'll show you an update on, GI, as well as in the liver. Now, how do SPMs work? Well, in this illustration, this is supposedly the postcapillary venial. I want to emphasize that SPMs limit the further recruitment of PMN, but stimulate the recruitment, non-phlogistic recruitment of mononuclear cells. So when macrophages encounter SPMs, they increase phagocytosis, resulting in the removal of apoptotic PMN, as well as microbes, and clear PMN from the sites. So the SPMs have proven in multiple in vivo animal models, experimental animal models, that they are not immunosuppressive. They counter-regulate the early initiators of acute inflammation, the prostaglandins. They regulate COX-2 expression, leukotrienes, PAF formation, counter-regulate the pro-inflammatory cytokines, increase IL-10, and you already heard this morning how important that is in the first presentation. They regulate NF-kappa B gene products, and ultimately lead to the regulation of edema. We have identified 5 separate GPCR receptors over time. You know well the studies with Mauro Peretti on the lipoxin A4 receptor, which is also shared by Resolvin D1, work with Lucy Norlin most recently in the knockout. And you can see that these receptors are expressed on different cell types, giving rise to selectivity that's been qualified with knockouts and transgenic mice, and most recently, the identification of the Resolvin D2 receptor and creating that knockout with Dr. Nan Chang. So if you want to read more about it, there's a nice review we did with Derek Gilroy and Chris Buckley. These receptors counter-regulate NF-kappa B, and they increase heme-1 oxygenase in many target organs. So now I'd like to emphasize our operationalization of lipid-mediated metabolomics work that we did in Boston when Jeff Stolle and Romain Kolos were present. They're here now, and I saw the operations set up here now at William Harvey. Very exciting, with the new mass specs in place and ready to go. We identify 6 diagnostic ions. I think this is a key point. A lot of other groups don't do this. We do the computational analysis, cluster analysis of these pathways to resolution, which we think are really critical not only in organ profiles, but in personalized and precision medicine in the future. So from this approach, what we learned is that we and others have shown now that many human tissues by mass spectrometry have been shown to produce pro-resolving mediators. I'll emphasize the breast milk here with recent work with Hildebrew Arnaudot when she was in our group, and she's now at the Karolinska Institute. And very recently, the profiling synovial fluids with Lucy Norlin, Mauro Peretti, and others, as well as the group in Australia, Barden et al. Some of the highest levels of the pro-resolving mediators are found in the placenta, which is quite intriguing, and as well in the lymph nodes. So our approach to human translation from the other side, which I'm sure is quite familiar to you, is since we have GPCR receptors, we made a series, and this is just an example with Resolvin D1, of receptor mimetics that we pursued in animal models and elsewhere to try to develop agonists to the pro-resolving actions. So in pursuing human translation, we have currently periodontal disease trial supported by NIH, which I'm serving as the PI and program director of, and this is ongoing at the Foresight Institute under the direction of Tom Van Dyke. With Resolvex, WHO awarded Navamepent as the name for the eye drops, and this trial was reported in phase 2, multi-center, double-blinded placebo. More than 232 patients were treated. This is the first demonstration of clinical efficacy with a pro-resolving therapeutic. So hopefully this will open up a new direction for treatment. And if any of you would like to enroll in this periodontal trial, here's the ad. We're halfway through this trial. So what about the relationship between the demands of the tissue on injury, resolution, bacterial invasion? Are there signals from resolving infectious exudate that act in tissue regeneration? And to address this with Jeff Stolle, we went back to our trusty mouse model of peritonitis, where you can see very rapid neutrophilic infiltration, and then the resolution phase illustrated here. Fax analysis to interrogate the pus that's produced. And this is an E. coli infection. And then we moved to a model organism, another one, the planaria, which you might be familiar with from your high school training. We can do surgical injury here. You can chop off their heads or tails, and they'll grow back very nicely in a very short period of time. And the animal committees are not that concerned yet, so we can move pretty quickly with the planaria. The nice thing is that on surgical injury, they grow back the tissue, and we took systematic extracts of the resolving exudates from E. coli infection to address this question: do we have mediators that can accelerate tissue regeneration? The first two that were identified, illustrated here through structural elucidation, they had a 14 series alcohol, so we knew that this placed them into the family of Marisins. Conjugated double bond system. This is a glutathione adduct that is 13 glutathionyl, 14 hydroxy. That's a long one to say, so we use MCTR1 for today. And 2 is right next to it. This is our other bioactive mediator that was identified. These are the means for structural elucidation to arrive at this structure. And we found these in human milk, of course in the mouse exudate, in human macrophages, and of course they stimulate the regeneration of tissue in planaria. What was very exciting for us is adding back into the peritonitis. And this is an E. coli peritonitis, which this is the resolution interval. It takes about 20 hours to resolve this dose of E. coli. When given at just 50 nanograms per mouse, this 20 hours is reduced, the resolution interval shortens to 10 hours. So we're very excited by this. And you would expect that this might lead to some reduction of neutrophilic responses, but in fact, this is an increase in E. coli phagocytosis and killing of the bacteria. So we pieced together the pathway illustrated here with Gestali. The epoxide intermediate has been confirmed, synthesized by Nikos Patasis, is an intermediate to MAR1, as well as the precursor to MCTR1 and 2, which have these biological actions. Since it's the William Harvey, we wanted to show you a ligation experiment. We use this hind limb model because this is an example of reflow injury to the lung and secondary injury to other organs that can be experienced in the OR during an operation of the limbs, for example. This is the sham lungs. Now you see very nice alveolar space. Reflow injury. This is a very good example of leukocyte-mediated tissue damage acutely, and when treated with just 50 nanograms per mouse, which is quite a low dose, you can protect that organ from second organ injury. Now, in assigning the stereochemistry, which we have recently for MCTR1, 2, we found MCTR3 in collaboration with Bernd Spur and his group in New Jersey. And so from the original publication, we're able to put together the stereochemistry of the entire MCTR pathway. Here we show the matching from synthetic. This is an LC-MS-MS chromatogram. These are the 3 synthetic compounds now, which helped us to improve the chromatography. This is the amount that's produced in mouse exudate. In sepsis patient blood, you see 2 and 3 present, and then human macrophages, 1, 2, and 3 are present. The other important point to make here is that MCTR1, 2, and 3, their MS spectra match the biologic and those of the synthetic compounds. So we use this to assign the double bond geometries and the chirality of the alcohols. Looking at the planaria, MCTR1, 2, and 3 are all bioactive, enhancing the regeneration, and you can see that MCTR3 increases its biological action, and this is reversed and seen in the cysteine-leukotriene pathway, where there's, in SRSA, there's a decrease in most of the biological actions from cysteine-leukotrienes 1, 2, and 3. So what about expediting or shortening the resolution interval? MCTR1 added back in here at the maximum of PMN infiltration in the peritonitis. It changes the resolution interval from 12 hours to 10, MCTR2 from 12 to 8, and now the synthetic MCTR3 from 12 to 9. So substantial reduction in the time. In this human macrophage experiment, you can clearly see the phagocytosis. This is real-time imaging. It's carried out by Nan Chang, who's here today as well. At 1 nanomolar, MCTR3 is quite potent, above that of E. coli alone or MCTR 102. And the image doesn't do it justice, but the fluorescence here is fluorescent uptake of E. coli, fluorescent E. coli. So when we went back and looked at infected mice now and looked into this organ here, it's spleen, even I can identify that this is a spleen in the lab, and we took exudates from the peritonitis, and you can see PCTR 1 and 3. So what's PCTR? Well, here's the structure, and since the alcohol group was at the 17th position with a triene, we knew this was related to the protectin family, and these are the conjugates that are produced, 1 and 3, from the epoxide intermediate related to the protectin biosynthetic family. So this is the pathway we've been able to put together, the protectin pathway, which are conjugated trienes, and the Resolvin pathway, which are conjugated tetraenes, formed from the 17 hydroperoxide intermediate. RCTRs 1, 2, and 3 on left, PCTRs 1, 2, and 3 on right here. And they are each bioactive, stimulating bacterial phagocytosis, killing, stimulating phagocytosis and clearance, and promoting tissue regeneration. Here is an example of the biological action on human macrophages, PCTR1 in black, dose response, PCTR2 in blue, PCTR3 in green, compared to protectin D1. This is all in nanomolar concentrations. These are the RCTRs on the right, shown here in black, RCTR1 and 2. So key in the biosynthetic pathways here was this epoxide intermediate, shown in red. We studied this and proposed this intermediate from oxygen 18 incorporation studies and found that there's a precursor to protectin D1. This was successfully synthesized by Trond Hansen and his group in Oslo. He was on sabbatical with us in Boston. And we've been able to show that this is converted to PCTR1 by macrophages of the M2 phenotype. And here is an example of the epoxide intermediate, the known stereochemistry, 16S, 17S epoxide, and this is trans-cis double bond geometry, and converted to PCTR1, and here is the appropriate LCM-SMS matching for the ions. But the biological action is what's very intriguing again for PCTR1. Here's E. coli clearance during infection in the exudate and in the spleen, enhancing macrophage biological action in both organs, as well as the killing and clearance of E. coli. What you can see here when we look at the resolution interval quantitatively, again, in the absence of any of the pro-resolving agents, the E. coli resolves in this case right here, and you can see a percent change with PCTR1 in this system going from 21 to 9, or more than 50% reduction. This is also the case for accelerated surgical wound healing, if you will, and this is an example, again, of the planaria with PCTR1. And we went back and looked at agonist-induced activation of cytokine arrays with a variety of cytokines and growth factors, and you can see very clearly with serum-treated Zymazan as an agonist to the macrophage. With PCTR on board, there's a substantial reduction in TNF-alpha, IL-8, IL-12, and there's even stimulation of a number of other key regulators, for example, like leptin and rantase by PCTR1. So the lion's share in the M1 macrophage is illustrated here in red for the SRSA, leukotriene C4, D4, and E4, which I'm sure you'll hear about from Professor Samuelson, who was also one of my mentors. And in blue, the summation of the three pathways I just illustrated for you now, the MCTR 1, 2, and 3, the conjugates for the protectins and the resolvins. Now, when we move to the M2 macrophage, you can see a dramatic shift where there's less leukotriene production in red and a substantial increase in blue, up to more than 70% of the new pro-resolving conjugates. So we have now, in wrapping up, illustrated for you today, for this William Harvey's celebration, 1, 2, 3 epoxide intermediates that are critical in producing pro-resolving mediators, as well as conjugates MCTR 1, 2, and RCTR 1, 2, and 3, and PCTR 1. Just want to remind you that the SPMs function to actively keep the inflammatory response within physiological boundaries of acute, to accelerate, the return to homeostasis. They do not block inflammation. There's no immune suppression. They facilitate the clearance of debris, and these conjugates directly promote tissue regeneration. So what we learned now is that there's a conserved chemical structural unit. This is an epoxide intermediate, illustrated here, which is key from essential fatty acids. In the case of arachidonic acid, the pro-inflammatory mediators and smooth muscle contractors, the leukotrienes are made, and from the N3 essential fatty acids, the pro-resolving mediators are produced. So we learned a lot from this surgical injury of the tissue regeneration of planaria. We can now know that from E. coli infections, there are mediators that are produced, as well as in human milk or in human macrophages, that help to regulate the inflammatory response and then directly act on tissue regeneration. So in conclusion, we have identified the Marisin conjugates, the protectin conjugates, and the Resolvin conjugates in tissue regeneration. They are abbreviated here. Here are their main biological functions that we know of today, and I hope that working with Jess and Roman, you'll discover many, many more new functions for these molecules. We have established their stereochemistry, 1, 2, and 3, and yes, that they're producing mouse lymphatics, and as we emphasized, infectious inflammatory exudates. So with that, I want to go back and emphasize the members of my group that contributed to this particular work. This is Hilda Brew-Arnodon. She's now at the Karolinska Institute. She worked on the milk I told you about. There's Jess Dali, who's here. Roman Kolis, who's here now, and have operationalized that LCMS-MS system. Jeremy Winkler synthesized with Tron Hunson's epoxide PCTR1. And then I'd like to point out our entire program project team, which is underway and busy studying these pathways now. So with that, I thank you very much for your attention.
It's absolutely fascinating, Professor Serhan. Questions?
Charlie, thank you for an excellent talk. You probably already know what I'll be asking you about. You've shown very nicely what different probable mediators are for connections between the host and
That's a lot for the first question. Okay. So I'll try and tease that apart. So the first part of, and I can tell you that there's not much that's been studied regarding the microbiota. So there's a lot of opportunities there in terms of the pro-resolving mechanisms. What we have learned is that certain primordial P450 systems in bacteria can produce the first intermediate in the resolving pathways. 18E was the first one identified, and so can 17E di-HET produced from DHA. So what does that mean? That means that there are bacteria in our gut that very likely metabolize the first step, which we can have trans-organism uptake in biosynthesis. So I believe that this is one of the pathways. In terms of adding back pharmacologically, resolvins do regulate the local microbiota. That's been demonstrated in the periodontium in a couple of studies, one from the foresight. But that's just at the beginning of the game. And I think that as we regulate the inflammatory response locally, we're certainly going to have an impact on bacteria. And I don't have a question. I don't have an answer for the last part of your question.
There's a question here and then.
That was wonderful. That's beautiful. Do you have any
Yeah. Okay, so Professor Samuelson is asking me for quantitative information. Of course, we want to have quantitative numbers to calibrate each of the components of the time course. And I didn't have time to show you this, but I did have a slide to address this. And at least in the... See, I get to show you all my slides now. The aspirin slide? Okay, right here. So this is the quantitative information, the type that we'd like to have during the time course of experimental acute self-resolving inflammatory response as pus resolves at the site. And this is a principal component analysis, and it's very cool. If we look at this in 2D, this is from the LCMS quantitative data. You can see very nicely in a peritonitis that we can identify all of the principal mediators and quantitate them. That's shown here in the loading plot. And what's, I think, is most fascinating is there's a clustering at time 0, 4 hours, 12 hours, 24, clustering of what? Clustering of the lipid mediators. They're changing the quantitative relationships at each point along the time course to full resolution. So yes, we have this. This has been submitted for publication, and hopefully it'll be published soon. And we do have quantitative data from plasma and serum in healthy individuals, and we're going about trying to do this time course of challenge work right now with Professor Derek Gilroy, who's in the back of the room there. And we do have some data coming along those lines.
Just to continue on from Professor Samuelson's question, so there's a changing time course of the expression of these different resolvins at different stages of the inflammatory response. So in your clinical trials, and when you're giving back resolvins or adding them into the environment, I noticed the trial that's underway, it's a local application. So what about in the chronic inflammatory situation? How do you see the field moving in terms of the resolvins being useful in the therapeutics of chronic inflammatory disease?
Right. You very rightly pointed out that in the ocular trial, that's an acute trial for resolving inflammation from dry eye. But in the periodontal disease trial, periodontal inflammation, the periodontists tell us this is chronic. This is a form of chronic inflammation. And so the data's not in there yet, but we're hoping to see counter-regulation of the excessive inflammation that goes on during repeated bouts. The way I look at this is failed resolution accelerated each time, so just short bouts of acute inflammation that accelerate. And that I think we can counter-regulate, and then the holy grail would be to get back tissue, and I'm hoping that they do see that.
And the PK/PD of these lipids, I mean, do they, I'm just thinking about long-term treatment,
Yeah.
How do you do it?
Okay, so that's a very challenging question. We want to think about this from two different perspectives. One is the physiology. At the site of inflammation, these lipid mediators are autophones. They're working locally, they hit their receptors, and then they're rapidly inactivated. And we have been working on the inactivation pathways, and that was I would say, the stimulus behind making stable analogs to metabolic inactivation. But pharmacologically, when you add them back, quite surprisingly, they're stable for a long time. Around in human whole blood, they seem to be protected from degradation by albumin and other serum proteins. And the PK that's been done on the eye for the eye drop is they have a half-life around 7 hours, which is surprising. Now, the one thing I should venture to say, that what we have learned experimentally is that this is a feed-forward system. Meaning what? Meaning that if you give a Resolvin, you upregulate its receptors at the mRNA level, is a feed-forward system. And the other thing we'd like you to think about is that if you hit the resolving pathway, the first die of the domino falls, but you don't have to have the compound there. It can still have action. And I guess the best example mentioned today is aspirin. Aspirin is not present at all where it has pharmacological impact, 14 days of the platelet life cycle afterwards. So we have a lot more to learn about PK because each one of them are going to behave differently.
Very exciting. Claudia.
Thanks for this excellent talk. I wanted to go back to the model of peritonitis.
Yeah.
You show that the resolution of inflammation corresponds or matches with the disappearance of nutrients from the environment, but also we call macrophages, I see. So are macrophages actively promoting the resolution? I think you hinted to that at the end of the talk.
Yeah, this is very astute of you. I didn't have time to really go into this ability. The pro-resolving mediators stimulate the M1 to M2 class switch. They are also responsible, as there are a number of mediators like the lipoxins, and then very recently Dr. Gilroy and his colleagues showed that some EETs can carry out this function, the non-phlogistic recruitment of mononuclear cells. So you have this going on dynamically. And yes, I think ultimately Derek's group, our group, have defined what we call the resolving macrophage, which is a specific phenotype that expresses arginase positivity. And I think that's most of the points that answer your question. Jess, do you have any comments, further comments? Derek, you're an expert on that. You want to add in? No? But on the break, we can talk about it more because it is, I think, the regulation of the neutrophilic infiltration is what we were originally after because we think that they are responsible for the havoc of excessive leukocytic recruitment and tissue damage. But the clearance is all the macrophage function.
Well, thank you very much, Professor Serhan. Certainly a very stimulating final talk for this session. I'm sorry, I know there are more questions.